Hey everyone, so today we're going to learn about mitosis. And you guys should already know about mitosis, but let's go through the PowerPoint really quickly. That way you guys can see all the different stages and learn all about them. There you go. So, mitosis is the process of cell division, which you know. First and foremost, there's a couple things you need to learn before we move on, and we need to talk about centrioles. Centrioles, I told you guys before you didn't need to know them, now you do. They are two tiny structures in the cytoplasm, and they make these things called microtubules, which are known as spindle fibers. Definition of spindle fibers are long strings that attach to the centromere and eventually pull sister chromatids apart. So they're really long, skinny strings of protein, and they attach to the chromosome, and they pull sister chromatids apart. You don't know what those words mean yet, but you will soon. So, where does information come from? So our DNA is the genetic information that codes for all of our traits and also makes sure that our cells divide. So why don't cells just grow forever? Why do we even need them to divide? And that's because we have a limited amount of DNA. And the size of the cell, if it got too big, it wouldn't work very well, which either you already know or you're going to find out soon from the lab that we're going to do. So a reason is the larger a cell becomes, the more demands the cell places on its DNA and the more trouble the cell has moving enough nutrients and wastes across the cell membrane. So the bigger you get, the harder it is to be a cell. So that's why we divide in the first place. So you already know a little bit about chromosomes. So chromosomes are the combination of DNA and protein molecules. So normally they're inside the nucleus in a soup, kind of, like they look like a bunch of noodles, and they're called chromatin. And then they condense into chromosomes, which are the X's that you guys are used to seeing. And a chromosome, by definition, is an organized structure of DNA and protein that consists of two identical sister chromatids when replicated, which attach at a central point called a centromere. Again, more language that we're going to get into more. Basically, a chromosome is a bunch of DNA in the shape of an X, and the two sides of the X are identical because we replicate our DNA, and they're exactly the same. So go ahead and draw your own chromosome. So make sure that you get this all down. So before replication, this is what a chromosome looks like. All these little guys are genes. So this might tell you what color your hair is. This might tell you how tall you are. This might tell you how long your fingernails grow. This might tell you what color your eyes are. And then we go through replication, and we have two copies of the same exact thing. Look, all the genes are the same. Okay? All of them are the same. And the two are attached at this thing called a centromere, right there. It's where the two sister chromatids are attached. So we have one sister chromatid and another sister chromatid, which are identical, and they are attached right there. And this is after duplication. So there's the whole cell cycle. It's the whole cell's life, and by definition, during the cell cycle, a cell grows, prepares for division, and divides to form two daughter cells, each of which then begins the cycle again. It's the definition of the cell cycle, and there's four phases: G1, S, G2, and M. So G1, S, and G2 are all part of something called interphase, which means between. So it's between cell divisions are the three parts of interphase, and then we have mitosis, the M phase. So DNA replication happens during the S phase of the cell cycle, and when DNA replicates, each chromosome gets a second copy of itself, which is identical, and enzymes in our bodies help to make the new DNA. And as soon as we replicate the DNA, we now have two copies of each chromosome, because we had one copy and we copied each of them, and so now we have two copies, and we call the cell diploid. Di means two. So we have two copies of each chromosome. So mitosis is the actual process of cell division. And mitosis occurs in all somatic cells, and a somatic cell is anything that's a non-sex cell. So that's any cell inside your body except for your sex cells, which are your sperm cells if you're a gentleman, or your egg cells if you're a lady. So all the other cells in your body. And it happens in four steps: prophase, metaphase, anaphase, telophase, and cytokinesis is kind of part of telophase. So prophase, we're going to start off with, it's the first and longest phase of mitosis. First and longest phase of mitosis, not of the whole cell cycle, just of mitosis. Takes up to 50 to 60% of the total time required for mitosis to happen. During prophase, these are things that happen. The chromosomes become visible. So they go from being that soup to being those X's, so we can see them now. Our centrioles, those two little structures, they're in one spot, but then they start to move to opposite sides of the cell. Our spindle fibers start growing from the centrioles, and the nuclear envelope, the space around the nucleus, disappears. So now the chromosomes are free inside the cell. So here's everything we talked about. Centrioles are moving, spindle fibers are growing. Chromosomes, we can see them. You can actually see the X's, and the nuclear envelope is beginning to break down. If it weren't, it would be not dashed lines, it would be solid lines. So go ahead and draw this. You can pause it and draw this where it says draw prophase. This is what real-world prophase looks like. So you can see how like the chromosomes are starting to condense right there. They don't look like noodles over here anymore. It's hard to see. If we had a better magnification, we could see it a little better, but that is prophase. Next is metaphase. What happens during metaphase is we have all of our X's, and they're going to line up right down the middle of the cell. X on top of X on top of X. And randomly, this happens totally randomly. And then spindle fibers, those things from the centrioles, come in, and they go and they attach to the centromere. So that's the middle part of the chromosome. They attach to the centromere on each side of each chromosome. So here's what it looks like. Here's our centrioles, they're on either side. They send out spindle fibers and they attach to the centromeres of each chromosome, which are lined up right down the middle at the metaphase plate. So the spot where they line up is called the metaphase plate. All the chromosomes are like, hey, come meet me at the metaphase plate. It's right here. This is what real-world metaphase looks like. So this is a real picture of a cell with all the chromosomes lined up right in the middle. They're all kind of, remember cell's 3D, so it doesn't look like X's, but they're all stacked up right there. And they're all attached by the spindle. Anaphase is the third step of mitosis. And now we're going to pull apart those sister chromatids. Remember, we have identical copies on each side, like this, and they're attached at the centromere. And the spindle fibers pull them apart, so now they're not attached anymore, and they're pulled to opposite ends of the cell. The cell begins to elongate, so it looks more like an oval than a circle, because we're pulling it apart. And this is the step where sister chromatids become individual chromosomes as they're separated. So here's what anaphase looks like. Go ahead and draw it. We have our sister chromatids being pulled apart. So here's one sister chromatid, here's another. They're attached at the centromere by the spindle fibers, and they're being pulled apart. This is what real-world anaphase looks like. This is probably the most distinct phase of mitosis if you're actually looking at it. Like you can actually see all of them being pulled apart. So the centrioles are right here, and they're being pulled apart. Lastly, we have telophase. Telophase is when we are starting to make two entirely new cells. It's the final step. The chromosomes begin to break down into the noodles again. They don't need to be the X's, the chromosomes anymore. And the cell is still pulling apart. So now the cell is actually going to start to divide. And that happens, so we have our cell, and it starts to pinch in like this. And the space right here is called a cleavage furrow. So that's starting to cleave. And the spindle fibers and the centrioles break down because we don't need them anymore. We only need them for cell division. And then our nucleolus and our nuclear envelopes start to reappear. So that's what happens during telophase. So go ahead and draw it. Here we see it again. The chromosomes are starting to break down. Remember, they're no longer X's because we just pulled them apart during anaphase. But now the nuclear envelope is back, starting to reform. And the centrioles are starting to break down. And the spindle fibers are gone because we don't need them anymore either. And right around here is the cleavage furrow. It's where telophase, where the cell membrane is starting to pinch in. And there we can see it. See how clearly defined the shape of the cell is right there. Then lastly, we have cytokinesis. So this happens simultaneously to telophase, same time. And what happens is cytokinesis means splitting of the cytoplasm. So we're actually splitting the cell into two daughter cells. And they have the same exact chromosomes. So see how identical these two cells are? Because they just underwent mitosis, so now we have two identical daughter cells. We're not going to do that. All right, that's all, guys. Go ahead and answer the questions and have a great break. Thanks.